Good morning. I hope you guys have had a good day here so far. My name is Matt Spitzer. I'm a PhD student in the Nolan Lab in the immunology program here at Stanford. And I'm going to be telling you a bit about the way that we generate reagents for CyTOF application. So hopefully for those of you planning on doing CyTOF experiments, this will be helpful. It's kind of the bread and butter of making sure that you have the tools that you need to ask the biological questions that you're interested in. So first, I'll go through kind of the overview of how we conjugate antibodies and how we assess their validity. So I'll start with a chemical overview of the coupling chemistry that we use to tag antibodies. I'll go through our conjugation protocol for how we actually execute that chemistry, talk a bit about how we titrate antibodies and validate them to ensure that they're actually binding to the target of interest, and then tell you a little bit about how we troubleshoot when things don't seem to be working as well as we might hope that they should be. And then I'll touch a little bit on some of the other reagents that we use for CyTOF application that are not antibody-based. And so in Sean's talk this morning, I'm sure he explained to you how you get, at least in theory, above 50 or so parameters using CyTOF. Currently, we're measuring 40 parameters on a single cell level, at least 40 proteins. And in order to do this, we take advantage of the fact that the lanthanides that we tag our antibodies with have multiple stable isotopes. So for example, europium here that has an atomic weight of 152, what that's actually comprised of is about a 50-50 mix of two different stable isotopes of europium, one that's 151 atomic mass units and one that's 153. And because of that distinction, we can actually label two different antibodies using europium metal, as long as we're sure that one of them has each of those two different stable purified isotopes. So this allows us to expand beyond merely the number of atoms and actually take advantage of the fact that they have multiple stable isotopes. And so when you look at the periodic table, what's surrounded here in blue are the atoms that we actually use in CyTOF application. So basically everything from a molecular weight starting at 102 atomic mass units all the way up to the end of the periodic table. We routinely use the lanthanides, which range in atomic weight from 139, which would be lanthanum, to 176, which is the highest atomic weight of ytterbium. So that's kind of the range that you're looking at on the periodic table. And to give you an idea of how we use these, the low end of the mass spectrum, we use palladium to execute our barcoding strategy that you'll hear more about tomorrow from Eli Zunder. But basically, the barcoding is a way that we can multiplex samples to stain them all in one tube, run them all on the instrument at the same time, and therefore obviate any technical variability between samples that we're really interested in comparing. In terms of antibody labeling, we use metals ranging from lanthanum down through the lanthanide series, mainly to label our antibodies, in addition to indium here. And then most commonly, to intercalate our samples to measure DNA content, we use iridium. So now I'll give you an idea of the way that we actually tag these antibodies. And the basic idea, as I'm sure you're all familiar from this morning, is that rather than tagging antibodies with fluorochromes and interrogating them with lasers, we tag them with these stable heavy metal isotopes, which allows us to discriminate these reporter species by mass spectrometry. And the way that we do this is by tagging our antibody to a polymer. And the polymer chain includes chelating groups, and those chelating groups bind our heavy metals of interest. And so the stable chelating chains are comprised of DTPA, or diethylene triamine penacetic acid. And that's the chelating group that actually binds in its core the lanthanide ion of interest. And so these chelating groups are really great for binding trivalent ions, 3+ ions, and those are the metals that, or the salt form of the metals that we use to label our antibodies. And importantly, it binds trivalent metals with a really high affinity. The KD is 10 to the negative 15, which is, or 10 to the negative 16 actually, which is actually like an order of magnitude tighter than biotin binds streptavidin. So we're talking about a really tight non-covalent interaction here that's essentially non-reversible. So under the conditions you would use for a mass cytometry experiment, these metal ions are not going to come off your chelating groups, which is fairly important because it means that we're not going to be swapping around signal between our antibodies that we're interested in using. And so typically using our conjugation strategy, we're labeling our antibodies with 1 to 300 metal atoms per antibody. It tends to be more on the 100 end of that spectrum. And then importantly, after we've labeled our polymer, we need to couple that polymer onto our antibody. And to do that, we use sulfhydryl chemistry. So there, as you may know if you've done any antibody derivatization, there are kind of two main ways that people derivatize or conjugate antibodies. One is using primary amine-based chemistry, and one is using sulfhydryl-based chemistry. We use the latter. And to do that, our polymer has a bismuleamide linker on the end of it. And that linker is sulfhydryl reactive, meaning it'll bind SH groups. And so in order to free up those sulfhydryl groups, we need to partially reduce our antibody, because sulfhydryls in proteins, at least, often form disulfide bridges. So they're not immediately available. So we have to break those disulfide bridge bonds in order to free up a sulfhydryl group, in order to let our bismuleamide linker react with it and to bind our chelator or a polymer onto the antibody. So I'll tell you a bit about how we do that. And importantly, this chemical reaction also essentially goes to completion, which means that under standard conditions, when you're labeling antibodies, you're not going to have unlabeled antibody sitting around afterwards. And so the first place to start when you're thinking about how to label a new antibody is choosing the antibody clone that you're interested in. So you want to identify the target that you want to measure, and then think about which antibody clone is most suitable. And generally, what we do in Gary's lab is to choose an antibody clone that's been validated for flow cytometry if one exists. So if you have an antibody that you really like for fluorescence-based flow cytometry, go ahead and use that same clone to label for CyTOF. This provides a really nice and easy means to compare the binding characteristics of that antibody after you've labeled it, since you can use the same reagent on a fluorescence-based cytometer as a primary conjugate. And it also generally means that the antibody species you're working with is robust and has pretty favorable binding characteristics for the types of assays that we're interested in using them for. Now, if one of these clones that people use for flow cytometry doesn't exist, it is still definitely possible to develop CyTOF-friendly reagents to measure different targets. It's more challenging because you don't necessarily have that gold standard of a fluorochrome-labeled antibody that works well for flow, but we do it pretty routinely. It does require that you find a purified monoclonal antibody or prepare one yourself. And in order to validate it, you'll want to be a little more stringent since it's not a common reagent. So you really need good positive and negative control conditions to ensure that the binding specificity is what you want it to be. And I'll get back to that in a little bit here. But first, I'm going to tell you a bit about the chemistry that we use and the reagents that you'll need to execute it. So there are three main components to labeling metals onto antibodies. And the first is that you need an antibody. We generally start by labeling 100 micrograms of antibody at a time, which is enough to allow you to recover somewhere between 100 and 400 tests of antibody for downstream application. You certainly can scale this up. And now in the lab, we pretty routinely label like 500 micrograms at a time if they're things that we're sure we're going to use a lot of. And it's important that it be carrier-free. So we don't want any other proteins sitting around in solution because they could react with the polymers that we're trying to label our antibody with. And importantly, BSA is probably the biggest one to avoid. It has a lot of disulfide bonds, a lot of sulfhydryl groups. You also need polymer. So we label our polymer to antibody in a 2-to-1 ratio, so 200 micrograms of polymer by weight, not by quantity. So 200 micrograms of polymer, which you can acquire from DVS Sciences, the company that makes the CyTOF, and this is actually now Fluidigm. I should change my slides. And then also a lanthanide solution, and we use trivalent metal salts that commonly come in the form of the lanthanide trichloride. And we keep these in stocks of 50 to 100 millimolar in solution in our lab. So those are kind of the three main essential building blocks that you need to label your antibody. You need the antibody itself, you need the polymer, and you need the metal. And then on top of that, there's a series of buffers that we use for the different steps of the antibody labeling. This is all on your slides there. So We use a loading buffer to load the metal onto the polymer. We use a reducing buffer to reduce the antibody to free up those sulfhydryl groups, a coupling buffer to combine the polymer with the antibody, and then a washing buffer to wash the antibodies. And these are all commercially available. We need a reducing agent. In the lab, we use TSEP, or tris carboxyethylphosphine, which is a pretty widely used reducing agent to break disulfide bonds. It's fairly mild. And then we also keep our antibodies in a stabilization solution. So this is just a list of the things that you'd want to have on hand if you're sitting down to start making reagents for yourself. That stabilization solution has some carrier protein, so it'll keep your antibody happy in solution, and as well as some salt to keep everything nice and buffered. And then in terms of raw materials, in order to do all of these buffer exchanging steps, we use millipore centricon columns, which are just small filter columns that you can throw in the centrifuge to do your buffer exchanges pretty easily. And then the other important thing to note is that you really want to use filter pipette tips when you're doing anything related to CyTOF to prevent metal cross-contamination, which can happen pretty easily if you don't have those filters. So now I'll tell you a little bit about some of the things that you just want to be aware of, kind of the most important things to keep in mind when you're going about labeling antibodies for yourself. And the first is that the concentrations in our protocol are really pretty important. And most notably, lanthanides, or metals in general, have a tendency to cause proteins to precipitate. And so if you add your antibody into a solution that has a pretty high metal concentration, you'll probably end up with a nice clump of antibody down at the bottom of the tube that you will never see or use. So that's one thing you really want to be sure to avoid. The second is, as I mentioned, the purification of the antibody. And this is also pretty important, that you don't have any carrier protein in solution with it. Otherwise, all of your polymer will more or less end up bound to that carrier protein and not to your antibody of interest. There are some carriers that don't give us problems, and those are namely ones that don't have disulfide bonds. So if they don't have the chemical group that our polymer reacts with, it's fine to keep them in solution. And then the last thing is the timing. Since we're reducing antibodies, we're destabilizing them, essentially. We're breaking bonds that are important for their structural integrity. And so we want to make sure that we're not subjecting our antibodies to these kind of harsh conditions for too long of a period of time. So the timing of the protocol is also pretty important. It's not one of those things in the lab that you want to leave sitting around while you go grab lunch and come back in a couple hours. So now I'll kind of go through a high level of how the chemistry works, and we'd be happy to provide people with a more detailed protocol to actually see all of the steps if you're interested in doing this yourself. But here's kind of a conceptual idea of what's happening. And so the first step, as I mentioned, is to couple the metals to your polymer chain. And that produces a polymer with chelating groups that have bound your trivalent metal ions. It's important to do this step first, as I mentioned, so that your antibody, the protein, isn't seeing such a high concentration of metal ions. So we let this reaction go to completion. And then at the same time, we take our antibody and we perform a partial reduction using tris carboxyethylphosphine in order to break those disulfide bonds and expose the sulfhydryl groups. And then lastly, we allow the polymer chain that has the bismuleamide linking group to react with the sulfhydryl group on the antibody. And that ends up giving you an antibody bound to a polymer that's chelating the metal ions that you want to use as your reporter species. I'm just going to go through this one more time so people understand it, because it's kind of the bread and butter of how all of this works. So the first step, preloading the polymer. Again, it's important to preload so that the protein doesn't see a high concentration of metal ions. So we let our polymer group with chelators interact with our trivalent metal salt in solution. Second, using a reducing agent to break disulfide bonds in the antibody, exposing sulfhydryl groups. And importantly, the disulfide bonds that we're actually reducing are the ones that sit in the FC portion of the antibody. They're actually more susceptible to reduction than the sulfhydryl groups in the FAB portion. And that's pretty important because the FAB, of course, is what's actually conferring the binding specificity to your antibody. So the conditions that we use are tuned in such that we're pretty much leaving the molecular interactions here alone, but we're reducing the disulfide groups up here in the FAB, where maybe we're not quite as concerned. And that provides us with the sulfhydryl groups that we need to actually couple our metals onto the antibodies. And then the last step is taking this partially reduced antibody, adding it to our polymer chain with the trivalent metals chelated, and allowing that reaction to go to completion. So 3 main steps here. And after we've performed that antibody conjugation, we're now left with a batch of protein that we hope is binding the trivalent metals that we want to measure on the CyTOF. And in order to quantify how much protein we actually end up with in our tube, we do this just by using a spectrophotometer, so like a nanodrop. It's pretty straightforward. You measure the absorbance of your solution at 280 nanometers, and the absorbance is a function of your concentration, so that'll tell you what your recovery was or what your yield was. We generally, when we label 100 micrograms of antibody, we generally recover about 60% of that, so about 60 micrograms. And then the next steps will be to titrate and validate your antibody to make sure that it is binding to the target that you're interested in, and I'll show you some examples of how we go about doing that. And then finally, to store your antibody, and to do that, we prefer to leave it in a stabilization solution with carrier protein and some azide to prevent microbial growth. We just store them at 4 degrees. So here are a couple examples of how you'd go about titrating antibodies. The first I'm going to show you is a really nice, clear, cut and dry example, kind of the most straightforward thing you could come up with. And so those of you who do immunology in the mouse, you'll know that the phosphatase CD45 is polymorphic. Some strains express one form, and some strains express another form. So there are these 2 different forms of CD45 that we call 45.1 and CD45.2. And so this gives us a really nice positive and negative control if we're trying to label one or the other of these isoforms. So in this particular example, I was trying to label an antibody that was specific for the CD45.2 isoform. And to titrate this antibody, I prepared a mixture of cells that had some CD45.1 cells and some CD45.2 cells. So a very clear positive and negative control population in the same tube. I could then use a counterstain antibody that I had already developed against CD45.1 to gate these cells into 2 main populations, those that did not express 45.1 and those that did express 45.1. So we have our positive control and our negative control. And then on those 2 populations, we could look at the expression of the new antibody that we were trying to measure, which is CD45.2. And as you'll see, only the cells lacking 45.1 have 45.2, whereas the cells expressing 45.1 have no CD45.2. So this is about as easy as a validation could get. You have a very clean, cut and dry example. And what you can see is you increase the concentration of antibody, you'll see a brighter and brighter signal on the CD45.2 positive cells, but the negative cells don't really change very much. So this is good. This is telling you that the antibody is behaving the way you would expect it to. And if you look at both of these populations together, you see a very nice bimodal distribution where that protein is either expressed or it's not expressed. So again, about as clear, cut and dry as you could expect it to be. Does that make sense to everyone? The logic, at least? Great. So here's another example. Labeling an antibody against CD3, which those of you who are immunologists know are expressed by T cells. So in order to assess whether a CD3 antibody is binding where we want it to, we might, say, stain a preparation of splenocytes, which we know will have T cells as well as other immune cells that are lacking CD3. So we have a nice inherently built-in positive and negative control population. T cells also express Thi1 or CD90, so we can use that as a nice positive control, as a counterstain, something that we know should work and label T cells. And T cells lack expression of B220, which is a canonical B cell marker in the mouse. So this gives us a really clean positive and negative control population. And looking at the expression of CD3, which is the new antibody that we're interested in titrating, you'll see that CD3 positive cells co-express CD90, whereas CD3 positive cells lack expression of B220. So this is nice. This is telling us that our antibody retains its specificity. It is selectively labeling T cells. And to go about choosing the appropriate staining concentration, we can look at the expression of CD3 across our T cells and see that as we increase the staining concentration from half a microgram per mil all the way up to 8 micrograms per mil, we really don't see an increase in the staining intensity of CD3 on our T cells, which tells us that we've kind of already saturated the target of interest even down here at 0.5 micrograms per mil. But what we do see as we start to increase the concentration is that the amount of CD3 that's sticking to our B220 positive B cells increases, which tells us that we're getting more and more background as we add more and more antibody. So this is exactly what you would expect if you're saturating the primary target. But it's important, I think, as an example of why adding more antibody isn't always a good thing. You want to really optimize your signal to noise and make sure that you're staining things at a saturated but not oversaturated concentration. And then here's just one more example, which is an antibody against CD44, which is an extracellular matrix receptor that people use often in the mouse to distinguish between different differentiation states of T cells, among other things. And this is kind of another example of how we might think about what concentration of our antibody to use, where as you add increasing amounts of our CD44 antibody, we do see a slight increase in the staining intensity, but it's really not very dramatic. And so, to say that a antibody is actually saturating its target, you would look at where this actually plateaus, and there's really not much of a difference after maybe half a microgram or 1 microgram per mil of staining concentration. So even though you are seeing some marginal increase, that's pretty much about where this is saturating. So hopefully that gives you a little bit of an idea about how we'd go about titrating and validating our antibodies, making sure that they are binding to the cells or the target that they should be, and also tuning in on the appropriate staining concentration. These are kind of the easier examples that I've showed you of cell surface proteins. Things get a little bit trickier when you're trying to validate antibodies against, say, a phosphorylated signaling epitope, which is what you'll be doing a lot of in the phosphoflow course. Obviously, that's why you're here, to learn phosphoflow. And so in order to measure those sorts of events, what we often will need to do is take unstimulated cells or cells that have been stimulated with some sort of perturbogen that induces the signaling pathway that we're interested in, and use those two conditions as our positive and negative controls in order to titrate antibodies of interest. So exactly what you use as those control groups varies depending on the biology of the target, but hopefully this gives you kind of a framework of how you'd go about thinking about that for your own biological questions. Briefly, troubleshooting. So oftentimes we'll label antibodies, well, hopefully not often, but from time to time, we'll label antibodies and they really won't be performing the way that we think that they should. And so what we often do in the lab as a first pass is to compare the binding characteristics of our metal-labeled antibody to either a purified antibody or a fluorescence conjugated antibody on a fluorescence-based flow cytometer. So we'll stain cells with metal-labeled antibody, purified antibody, or primary conjugate, and then use a secondary fluorochrome-labeled antibody in order to measure the staining intensity of our metal conjugate, run those samples on a fluorescence-based machine, and if we still see signal, basically that's telling us that the binding characteristics of the metal-labeled antibody remain intact. The problem wasn't that you denatured the FAB or that you let the reaction go too long or you used too much reducing agent and destroyed the binding specificity. It gives you kind of a nice way to tune in that the problem really was getting the metal onto the antibody or getting the polymer onto the antibody, but the actual antibody clone itself is still functional, which is really important because one of the reasons why our antibodies can fail is that maybe the FAB is a little bit more susceptible to the reducing agents that we use, and it's not an ideal clone to use for metal labeling. So that's generally our first pass. Check the staining on fluorescence using a secondary antibody. And if it turns out that you did denature the antibody and it doesn't have the binding characteristics that you would like it to have anymore, what we usually do is just give it a second shot. Label it one more time, see what happens. If the same thing happens, then we have some alternative labeling strategies that we can use to still measure that target. And those include things like a biotin streptavidin system that you're probably familiar with if you've done a lot of flow cytometry, where you can take a biotinylated antibody and use streptavidin coupled to a fluorophore. In this case, we use streptavidin coupled to heavy metals, but the same basic concept applies. Or the other thing that we can do in the lab is to use a fluorophore conjugated antibody. So you could take, say, the antibody that you're interested in measuring on a common channel like PE or APC and use that antibody, and then come in with a secondary antibody that's metal labeled targeted against the fluorophore. So that's another kind of trick to get around using clones that don't seem to be performing that well. Now I'm going to talk a little bit about some of the other reagents that we use. One of the other tools that we can use are nanoparticles. And the one that we use pretty conventionally now are the quantum dots, which are cadmium-based nanocrystals. And cadmium notably has a molecular weight that allows us to measure it on the CyTOF. It's in the 100 and teens. And so we can actually stain cells with different quantum dots that have more and more cadmium metal. And this is against the same target. And you'll see that the larger quantum dots give us a brighter signal on the CyTOF because they have more cadmium metal, which is what we're actually measuring, whereas the smaller ones give us a lower signal. So we generally use Q.605 or 655 if we're going to try to measure something using a Q dot on the CyTOF. And nanoparticle development is one of the areas of interest in the lab to try to kind of expand the number of metals we can use. We're turning to this kind of a strategy because we can get so much more signal by packing in a ton of metal atoms into a nanoparticle. And this is just an example of what one of those might look like, where here we're using, actually Sean and Aaron in their science paper that was published a couple of years ago, kind of describing the CyTOF technology, used a Q dot against CD3 in their panel, and they measured the signal on channels 110 to 114, which are the different stable isotopes of cadmium. The Q dots, of course, are not isotopically pure, so they have a mix of all of these different stable isotopes. And you can see a nice CD3 stain on their samples using that reagent. We also commonly use cisplatin in the lab as a viability stain, which was published in Cytometry, Feinberg et al. A couple of years ago. And this works pretty similarly to like a fixable live dead stain that you might use for fluorescence-based flow, like aqua or live dead blue. Basically, cisplatin will translocate very quickly across a compromised cell membrane and bind to sulfhydryl groups or free sulfur groups. Whereas in live cells, cisplatin does get actively transported across the cell membrane, but it's much, much slower. So we'll pulse in this reagent for 60 seconds and then quench with a high concentration of protein in order to selectively label dead cells for CyTOF. And then some additional reagents that we use. We often in our experiments will use a DNA intercalator. You'll do this in your experiment this afternoon. And the idea is to provide a common signal to all of the cells that are in your CyTOF experiment. So we don't have light-based scatter on the CyTOF because we don't measure light. So kind of instead of that, we often use a DNA intercalator in order to get some signal of where a cell is and what a cell looks like. Even if you don't have an antibody in your panel that would specifically measure that cell, we can still detect its existence by the fact that it has DNA. And so that intercalator that we often use is iridium-based. And Sean probably talks a little bit about that. And then another thing that we use commonly are the barcoding reagents that Eli will talk about coming up. So hopefully that gives you an idea of how you might go about starting to develop the tools that you would want for your own CyTOF experiments. I know it's very technical, probably pretty dry. Hopefully I didn't put everyone to sleep. But if you have questions, feel free to ask them now or at any point during the course. And in terms of who I am in the lab and what you might be interested in talking to me about, I'm kind of the mouse immunology guy in the lab. So for any of you who do that sort of stuff, if you're interested in chatting, I'm obviously around all week. So yeah, I'd be happy to take any questions, if there are any. Yeah.
So I've been labeling some antibodies in my lab, and I've seen some troubles with 139. I just want to know if you saw the same issues as well.
Yeah, so we don't really have problems with 139, but I know that DVS has had some problems labeling it. So I'm wondering I don't think we fully resolved why that is. We use it pretty routinely, but it might be that different metal preps, the different salts just don't work as well for the coupling. But I know you're not the only one who's had that problem, although I can't explain why you're having it.
Yeah, 'cause I talked to them recently and they had the same explanation. I just want to know if you guys saw something similar. I also have another question too. So you said you labeled 50 micrograms at a time.
Yeah.
Is there any changes to the protocol at all?
Yeah, there are some changes, and we can chat about that. It's mostly just scaling things up, but there are a couple things to tweak. Mostly, you just want to do a lot more washes because the volume you're resuspending things in is. Yeah, exactly. Yeah. Yeah. Sigma, because it's monoisotopic. Yeah. Yeah, I was just made aware of that pretty recently, so we haven't done a ton of troubleshooting, but yeah, hopefully we'll figure it out. Yeah.
And affinity purified polyclonal antibody could also work in theory?
Could definitely work, yeah. Generally, we stick to labeling IgGs. IgMs people sometimes use for flow cytometry, but as you know, IgMs are pentameric, and oftentimes they bind based on their avidity rather than their affinity. And with the reducing chemistry that we use, you're going to break apart that pentamer into monomers. So sometimes the individual IgM monomers will still have high enough affinity to bind. There are definitely examples of that in the lab of reagents that I use routinely that are IgMs that still work. But in general, affinity purified poly IgGs are fine. Yeah, for sure.
Do you guys exclusively use the linear X8 polymer? Or I feel like you guys early on also had a dendrimer that you could use?
Yeah, we do. So generally, we'll, at first pass, if we're labeling something new, use the linear X8 polymer. Sometimes the more globular polymer gives you slightly better binding characteristics, but it seems to also have higher background in general. So your signal to noise for most things will be a little bit worse. But in general, if you're having trouble seeing signal with the linear X8, it's worth giving it a shot with the DN3, which is their dendrimer, and seeing if it looks any better. But at first pass, we always use X8. Great.